I'm Shannon Mumenthaler. I am the Chief Translational Research Officer at the Ellison Medical Institute. I also hold faculty appointments at the University of Southern California in the departments of medicine and biomedical engineering. So we all know that cancer is a multifaceted disease. It's highly complex, and because of that, we have a lack of understanding in terms of tumor progression and also treatment response. And so our goal is to really better understand the interactions between the tumor and the surrounding microenvironment, how that microenvironment really supports the cancer cells, how we can ultimately target that and eventually prevent cancer from spreading. And so that's really where our research is headed. Microphysiological systems, in particular organs on chips, are really a paradigm shift in model systems and our ability to study very complex diseases such as cancer. And so we're really incorporating that into our suite of model systems. Number one, the fact that we really can incorporate multiple different cell types together in a tunable fashion allows us to study those unique contributions of the microenvironment to cancer spread, and then ultimately allows us to start to think about how we can treat that in the future. So I think especially when we're thinking about cancer invasion, we need to be able to look at different compartments of the tumor and look at tumor cells that are in the epithelial compartment. And as they start to leave that primary site and intravasate into the vascular system, the Organs on Chips platform really allows us to look at those intricate interactions and really that dynamic process where other model systems are unable to do that to that same degree. In addition, the fact that we can start to incorporate human-specific cells, and really it's not just the human tumor cells, but really the human microenvironment within these systems is also extremely powerful. And then in our case, we're combining patient-derived tumor organoids with the Organ on a Chip platform. And so really bringing two models together to allow us to study patient heterogeneity so we can make these personalized models and really look at the variability across different patients in terms of spread and ultimately in terms of treatment response. I think one of the big advantages of these models is the fact that we can actually uncover novel targets. And so I think from a drug discovery standpoint, we really will be able to start to look at new targets and ways to actually develop therapies against those targets and test in a more complex microenvironment. One of the challenges we have even now in terms of the various different drug modalities that we're looking at in cancer is that we're often testing those in animal models or in very simplistic in vitro models, and we're missing the mechanisms of action of these therapies. And so having an ability to kind of bring in organ on chips within the pipeline of model systems to test, I think will help us feel more comfortable and confident in terms of how we can move these new therapies through the pipeline.